Okay, this next segment of notes is all about DNA replication. So you know all about DNA and its structure with the phosphate deoxyribose sugar and the nitrogenous base. So now we need to go through how do we make new DNA. So we're going to go step by step through the nuts and bolts of replication. First of all, replication is an easy way, or the way of saying that we are copying DNA. We are replicating, making a new copy of DNA. So the very first thing is that we have a protein called helicase that unzips the DNA strand. So this is what helicase looks like. It drives right down the middle of that DNA strand and breaks those hydrogen bonds in order to pull the DNA apart. The second step is that polymerase binds to those pieces of DNA that are exposed because helicase unzipped them, and starts to make new strands by putting nucleotides that are floating in the nucleus, so A's, T's, C's, and G's, adenines, thymines, cytosines, and guanines, that are floating in the nucleus onto the exposed strand of DNA. And that is where we follow the rule A with T, C with G, and the other way around, T with A and G with C. And that's what polymerase does. It's the matchmaker of those different nitrogen bases. The third step of replication is when the process is totally finished and helicase has unzipped it, polymerase has added the new bases back on, and the DNA is closed back together. What we end up with is two identical DNA molecules that are semi-conservative. What does that word mean? So semi-conservative means that each molecule has one original strand and one new strand of DNA. So one that came from the piece that was copied and one that came from the piece that was assembled using the polymerase. And it's important to remember that it is semi-conservative, because what that means is that we're able to maintain the integrity or the DNA sequence a little bit better, because half of it's old and half of it's new. So we're able to make sure that there are not too many mutations by creating one entirely new strand of DNA that then goes and copies itself over and over and over again. So it is semi-conservative in that one half is original and one half is new. So this is kind of what it looks like. We didn't talk about primase, but helicase is down here in the purple on the right. It has unzipped the DNA molecule, and now we have polymerase that comes along and it's adding new base pairs and making our new strand of DNA. So what you'll end up seeing if this were to continue on, you would have an original strand that is blue and a new strand that is red. You would have a new original strand that is red and a new strand that is blue on the bottom half of the picture. The important thing to remember with DNA replication is it's always A with T, C with G. At and C with G, okay? Never ever forget, in DNA, A always goes with T, C always goes with G.